Greetings and welcome to the United States Transhumanist Party Virtual Enlightenment Salon. My name is Gennady Stolyarov II, and I am the chairman of the U.S. Transhumanist Party. Here we hold conversations with some of the world's leading thinkers in longevity, science, technology, philosophy, and politics. Like the philosophers of the Age of Enlightenment, we aim to connect every field of human endeavor and arrive at new insights to achieve longer lives, greater rationality, and the progress of our civilization. Greetings and welcome to our virtual Enlightenment Salon of November 6th, 2022. I'm pleased to bring to you today an illustrious panel discussing some of the cutting-edge advances and scientific research into aging and longevity and how we can improve our health and fitness. But before I introduce the panel, I would like to note this is our 1-year anniversary of our partnership with KGRA Radio, which has helped us bring these virtual enlightenment salons to a broader audience. So I would like to thank KGRA and our friend Bill Skywatcher behind the scenes for always doing such an excellent job in helping us coordinate these virtual enlightenment salons. We have been streaming the virtual enlightenment salons now for 2.5 years, and the latest of these years has been in partnership with KGRA. So now to introduce our panelists, we have our Director of Visual Art, Art Ramon Garcia. We have our Legislative Director, Jason Geringer. We have our Director of Longevity Outreach, Ben Vollweg. We have our Biotechnology Advisor and Founder and CEO of Sierra Sciences, Dr. Bill Andrews, and our two distinguished guests for today, who will be giving the introductory presentation, are Dr. Dudley Lamming and Dr. Adam Konopka. Dr. Dudley Lamming is a faculty member of the Division of Endocrinology, Diabetes, and Metabolism within the University of Wisconsin Department of Medicine. He is a fellow of the American Aging Association and the Gerontological Society of America, a recipient of the Gerontological Society of America Nathan Schock New Investigator Award, and the American Physiological Society and Endocrinology and Metabolism Section New Investigator Award, and an editorial board member of several peer-reviewed scientific journals. Dr. Adam Konopka is a faculty member in the Division of Geriatrics and Gerontology within the University of Wisconsin Department of Medicine. He is an editorial board member for the Journal of Applied Physiology and a member of the Gerontological Society of America, American College of Sports Medicine, and American Physiological Society. Dr. Konopka has been selected as a reviewer for several journals, including the Journal of Gerontology, Biological Sciences, Aging Cell, Geroscience, Science Advances, Scientific Reports, and Journal of Physiology. So welcome to both Dudley Lamming and Adam Konopka. It is a pleasure to have you join us today, and please begin with your presentation.
It's a pleasure to be here as well. Thank you for having us. So I thought today we'd probably focus on some of Adam and my shared interest in signaling through the mTOR protein kinase, which is familiar to many people as something that's inhibited by a drug, rapamycin, and that's gotten a lot of attention in the press and online as something that might be relevant to slow or prevent diseases of aging. So to take a step back, something that's been known for about a century now is that eating less can promote healthy aging. And this is called calorie restriction. Typically speaking, this is something that is done all the time in rodents. It's been done in higher species as well, including non-human primates. Here on the left is a study from Rick Weindrich, formerly of the University of Wisconsin, showing that if we look at mice that have free access to food or ad libitum fed animals, they have a certain lifespan, but restricting it by various degrees can significantly extend that lifespan. And the same is true in non-human primates as well. And so importantly, it doesn't just extend the lifespan of these animals, it really extends the health span. So animals that are calorie restricted by anywhere from 20 to 40% typically are hungry for sure, and definitely interested in eating food when it's made available. But they have lower rates of all sorts of age-associated diseases. And these include diabetes, cancer, cardiovascular disease. And in models that get these diseases, Alzheimer's disease and other things related to cognition as well. And so calorie restriction is really a powerful intervention in the laboratory. And there are definitely some humans who are following this regimens, although we don't know for sure that it works in humans as well. But of course, in our society, we're surrounded by food virtually at all times. And so we're really interested in trying to find ways to mimic the benefits of calorie restriction without having to have such an incredible amount of willpower that we would be able to restrict our calories by so much. So one thing that is key, we believe, to the effects of calorie restriction is a protein kinase known as mTOR. mTOR complex 1 is essentially a giant signaling integrating hub for all sorts of things related to nutrient signaling. And mTOR directly or indirectly senses things like the level of amino acids and protein, the amount of glucose, and things like cholesterol and lipids as well. And so when all of these are present, mTOR complex 1 turns on pro-growth signaling. And so this helps tell cells that has the availability to make lots of the materials needed for cell division or organismal growth. And so many different cellular processes are regulated by mTOR. And, just a few are listed here. This is one of these slides that's just sort of supposed to tell you that tons of things are regulated by mTOR. But basically, when mTOR is on, when nutrients are abundant and the organism is telling cells that it's okay to grow, mTOR complex 1 regulates the synthesis of proteins, ribosomes, which are the cellular machinery that makes proteins. It makes various different kinds of nucleotides so that DNA synthesis can occur and cell division can occur. And it does other things like store lipids and suppress autophagy. So lots of different cellular processes are regulated by mTOR. And in fact, in the lab, we often joke that pretty much everything is in one way or another downstream of mTOR. Now, the reason that mTOR is sort of so exciting to a broader audience than just scientists is the effects of a drug called rapamycin. So rapamycin is a molecule that was originally found on Easter Island, or as the natives called it, Rapa Nui. And it was found in a soil sample as something that inhibited growth of organisms. And to make a long story short, it eventually became commercialized on one hand as an immunosuppressant because it suppresses the immune system. It tells T cells and so on not to proliferate in response to antigens. This is useful for organ transplantation. But there was some thought that maybe this could be useful from an aging context as well. And it turns out there are a lot of suggestive data that says that mTOR signaling overall promotes not only cell growth and organismal growth and development, but also accelerates aging. And so mTOR signaling seems to be at a higher level than optimal in all different sorts of cell types, particularly we know the most in rodents. So this is a study done by the NIA's Intervention Testing Program. That's the National Institute on Aging. And they do unbiased essentially tests of a number of different molecules each year that are nominated by scientists. And what you can see here is that rapamycin was dosed to these animals in this particular study starting at about 9 months of age. What you can see is there is a significant extension of both male lifespan and female lifespan. Now, this has been studied extensively by many different laboratories over the course of a number of different experiments. All the ITP experiments are done in blinded fashion. Many other labs have duplicated these effects as well. And so when rapamycin is dosed to animals that are young or animals that are old, when it's dosed in low doses or dosed in higher doses, the sort of universal effect seems to be that mice and a few other model species live longer when they're treated with rapamycin. And so there's a lot of interest in saying, well, this is a molecule that certainly could be given to humans for anti-aging purposes. Might it protect them from age-related diseases? And in mice, rapamycin protects from a number of different age-related diseases, particularly cancer, but has shown benefit for many different things as well, including Alzheimer's disease. So why shouldn't we all be taking rapamycin? So we don't know much yet about what rapamycin would do at sort of low doses in humans. What we do know is what the lessons from high immunosuppressive doses in humans are. And this is because most humans who take rapamycin are taking it because they have transplanted organs or they have one of a few different genetic diseases or cancers for which rapamycin has been shown to be useful. So these are high immunosuppressive doses. And rapamycin is an immunosuppressant, and so people who are taking high doses get increased infections. There's also a variety of dermatological events, particularly some sort of ulcers that are sort of similar to canker sores. There can be hair loss and rashes. There are metabolic consequences, and that's sort of how I originally got into the rapamycin field, trying to understand those. And so some of these can be very serious, and some of these at high doses. So in one study where people were taking this for tubular sclerosis complex, a genetic disorder where you have high mTOR signaling and you need to take a lot of mTOR inhibitor, some people got infections, about 15 of 18. So the vast majority had infections. Some of those required going to the hospital. And other trials have shown lower rates of severe events, but they definitely do occur. Metabolic consequences also occur in these people taking high doses of rapamycin for long periods of time. And these include hyperlipidemia and problems related to glucose tolerance and insulin sensitivity. There's even an increased risk of new onset diabetes. How great that is not really understood. So what about translating this to humans? So one thing that we know for sure is that the adverse events are time and dose dependent. But we don't know about benefits. We think that low doses of rapamycin and its analogs, such as everolimus or temsirolimus, are likely much safer, but we don't know how safe and how efficacious they might be, even in rodents. And so that's sort of a gap in our knowledge, and we definitely don't know how safe and efficacious they might be in humans for disease of aging. One set of studies by Joan Manick and colleagues at a couple of different companies found that lower doses for 6 to as many as 16 weeks had very few, if any, side effects. And so that type of data suggests that low doses might be perfectly tolerable, safe, maybe even rejuvenative, helping to restore tissues and without adverse side effects. But we really don't know. And so, for instance, another study that studied elderly people or over the age of 65, very small numbers of people, but about half of them had side effects with rapamycin for about 2 months. And the effect on glycated hemoglobin, which is sort of a readout for diabetes, was very small, but triglycerides were elevated, but with about 40%. But again, these are all very small studies. With the exception of the studies led by Joan Manick that showed that there are very few side effects at these lower doses. And we think that there might be personalized medicine implications too. We don't know for sure, but work that has been done in rodents definitely suggests that genes and diet, and you can imagine pre-existing diseases as well, is likely going to impact our response to rapamycin. Adam, do you want to take over, or do you want me to keep going?
You can keep going. I think this is more your data.
Sure. So this is some glucose tolerance tests that were done in mice. Basically, if you look at mice that are treated with either vehicle or rapamycin, and it doesn't really matter whether it's via IP injection or it's middle-aged mice given rapamycin in a diet, or older mice given rapamycin in a diet, universally you get glucose intolerance. That's not true in humans, as far as we know. This is not a universal response in humans, but some humans do have this type of response. And what we found in mouse research that I did when I was a postdoc was that this is really an effect on hepatic gluconeogenesis. So rapamycin actually has effects in multiple different tissues, not just the liver. It affects insulin sensitivity in muscle and adipose tissue, maybe other tissues as well. But what we found in mice was that it actually doubled the rate of hepatic glucose production by the liver. And the livers of those mice that were treated with rapamycin chronically at a relatively high dose for about 3 weeks, they became much less insulin sensitive. So this is an experiment where we test how insulin sensitive the liver is. In the vehicle-treated mice, about 70% of hepatic glucose production shuts down when we give the mice insulin, but only about 40% does in the rapamycin-treated animals. Remember, those animals are producing more glucose to start with. And so overall, this seems to be a very important effect. Now, why this was so interesting was that it sort of gave us a handle to try and understand the mechanism of some of the side effects of rapamycin. And so we talked about mTOR complex 1 as a central nutrient hub, but there's also a second mTOR complex 2. And mTOR complex 2 is actually involved in transducing signaling from the insulin and IGF receptor downstream to many different processes, including mTOR complex 1. And something that had been investigated a little bit previously was that rapamycin, when given chronically, not only inhibits mTOR complex 1, but also inhibits mTOR complex 2. And so what we were able to show was that in mice that are given rapamycin chronically, we get disruption of both mTOR complexes. Now, this is a Western blot. This is the type of blot where we're looking at different proteins. And we immunoprecipitated mTOR with an antibody against mTOR, and then probed for its association with either Raptor, which is a protein component of mTOR complex 1, or Richter, which is a protein component of mTOR complex 2. And what we saw was that in the rapamycin-treated animals, we were unable to really pull down very much Raptor or Richter, but we pulled down a lot in the vehicle-treated group. And so this told us that, contrary to our initial expectations, this rapamycin, even though it's a very targeted molecule, it really only has one target at low concentrations. But when it builds up, particularly in vivo in mice, it can inhibit the second mTOR-containing complex as well. And so overall, the effects of rapamycin on hepatic insulin resistance are mediated by this disruption of hepatic mTOR2. And we tested this using genetic models where we knocked out Richter either in adult mice or specifically in the liver of young mice. And a number of other groups, particularly Brian Kennedy's group in 2015, showed that not only is this true in liver and white adipose tissue and skeletal muscle, but also in heart, pancreas, spleen, lung, virtually every tissue. And so mTOR complex 2 has a whole bunch of different downstream processes. And not only, we think a lot of them are mediated by AKT. Michael Hall's lab and Andreas Sukas have done a lot of work showing that some of the effects on glucose tolerance are related to inhibited AKT signaling. But there are tons of other substrates that are also regulated by mTOR complex 2. And I think overall, we tend not to know too much about that, although our knowledge of mTOR complex 2 has been expanding for years now. So a good question is, does rapamycin promote longevity via inhibition of mTOR complex 1, mTOR complex 2, or both? And we tested this using various genetic mouse models. What we found was that while inhibition of mTOR complex 1 genetically or with rapamycin extends lifespan, when we knock out Richter, which is a component of mTOR complex 2, we get shorter lifespan. So here we saw a 40% decrease in male Richter heterozygous mice. If we made a whole body knockout where we induced it in adult animals, there was again sort of a 40% or greater effect on median lifespan, where the mice are short-lived when they lose one or more copies of Richter. And this is true in other studies as well. This is a more recent study looking at Richter in the liver. Here's Richter in the hypothalamus. In all of these cases, we see animals that are living shorter and that are more frail. And so by knockout, what I mean is we are using genetic techniques to delete this one essential component of mTOR complex 2 named Richter, specifically in the case of the liver, in the hepatocytes of the liver. In the case of a hypothalamus, this is using a driver that's specific for some of the neurons in the hypothalamus. Same thing with adipose tissue. If we look at that, we see that there's an overall effect of Richter loss on lifespan as well. The animals live shorter when we inhibit components of mTOR complex 2. And so overall, we've come up with a model, and this model is that acutely rapamycin inhibits mTOR complex 1, and normally mTOR complex 1 signaling limits lifespan. And so by inhibiting mTOR complex 1, we can get effects where we extend lifespan and extend health span as well. But there's also this chronic effect of rapamycin where we inhibit mTOR complex 2. And if we inhibit mTOR 2 chronically, we get negative effects, particularly on male lifespan, maybe not so much on female lifespan, but we do see that sometimes now. And we get limiting effects on health span. And so depending on the tissue, mice lacking mTOR complex 2 tend to be fatter, frailer, as well, and have impaired insulin sensitivity compared to control mice. So some people, I should say, believe that signaling mTOR complex 2 inhibition is bad for longevity, is sort of too simplistic a model. And I will say that that probably is true, but the vast majority of evidence suggests that the more mTOR complex 2 activity you have, the better. And in fact, this has been shown in flies as well now by Wei Bai's group, that overexpression of Richter, so increasing mTOR complex 2 signaling in Drosophila, extends their lifespan. That hasn't been shown in mouse yet as well, but overall, there's a lot of positive evidence there. And the evidence that mTOR complex 2 inhibition can be beneficial is fairly limited. There's a little bit of evidence that under certain environmental conditions in C. elegans, which is a small worm, that could be beneficial. But in mice and in flies, there tends to be tons of data that mTOR complex 2 activity is good and that inhibiting it is bad. So can we get the good, this mTOR complex 2 inhibition, without the negative effects that we see? And so one thing that I've noted is that mTOR 2 inhibition only occurs chronically. And that's due to mechanism. Basically, rapamycin, when it enters the cell, binds to a protein called FKBP12, and that binds to mTOR complex 1. That region that it binds to on mTOR is accessible sort of on the surface of the protein complex. But mTOR complex 2, it's buried, and so it can't actually bind to mTOR complex 2. So what scientists think happened is that FKBP12 and rapamycin bind to newly synthesized mTOR and sort of prevent it from ever assembling into mTOR complex 2 in the first place. And so that's going to require time. And so you need high concentrations of rapamycin available for a long time. And so that suggested to us and others that an intermittent dosing of rapamycin might be beneficial. And so here's an example of that in mice. Here we tested and we found that dosing rapamycin every 5 days really more specifically targeted mTOR complex 1 and had fewer effects on mTOR complex 2, and yet still extended lifespan. And this is starting at about 19 months of age. So you could see even when the mouse is fairly old, you get good extension of lifespan with an intermittent treatment with rapamycin. There are a lot of people exploring intermittent rapamycin on their own in humans right now. And I think Adam will tell us about his ideas on that as well. So this particular regimen avoids the effects on glucose metabolism, so the mice don't become glucose intolerant, but still does have some effects on immunity. So it's not by any means a cure-all to have this intermittent dosing. We don't know if it's more or less efficacious in daily dosing either. Some rapalogs seem to have a reduced impact on glucose tolerance. So everolimus and temsirolimus, when we dosed it to animals, we found that it impaired glucose tolerance less than rapamycin did. They still had about equivalent effects on immunity, though. And that leads us to the question of whether there might be rapamycin analogs that are selective for mTOR complex 1. And so this is a drug that's developed by a company that has supported my lab, and I'm on their scientific advisory board. But this particular molecule, DL001, inhibits mTOR complex 1 quite well. It has similar IC50s as rapamycin. But when we look at mTOR complex 2, it's less efficient at inhibiting mTOR complex 2. And overall, if we calculate how much more selective it is, it's about 40 times more selective for mTOR complex 1 than rapamycin. And one thing that's really cool is that we could see this in vivo as having less of an effect on mTOR complex 2. Here's a very similar IP experiment to the one we did before. And you can see that if we look at rapamycin breaks up the association between mTOR and Richter, but DL001 does not. And we see in vivo that although DL001 inhibits mTORC1 signaling quite well, it doesn't have effects on glucose tolerance. It doesn't have effect on circulating cholesterol or lipid levels or free fatty acid levels. These are all mouse data, so you can take that for what it's worth. We don't know what it might do in people, if anything. So, in conclusion, rapamycin has negative side effects. Some of these may potentially limit its use for disease of aging. We just don't know yet. And many of the negative effects of rapamycin, we believe, seem to be mediated by off-target disruption of mTOR complex 2. In mice, if we genetically inhibit mTOR complex 2, we get mice that are less healthy and have a shorter lifespan. And at least in short-term experiments, we can avoid some of the effects on mTOR complex 2 by either dosing mice intermittently or with compounds that might be more specifically targeted to mTOR complex 1. And I'll turn it over to Adam now.
All right, yeah, thank you, Dudley. That was great. So now I'm going to kind of just unpack some of what Dudley's mentioned before and really highlight what rapamycin has been shown to have improvements on key physiological functions that are related to aging. And so, Dudley, if you just want to hit, I think probably about 4 or 5 times to move that forward. And so, really, some of the key findings with rapamycin is first that when dosed appropriately, or at least acutely, rapamycin can actually improve peripheral insulin sensitivity in young men, so in humans. Next, when rapamycin is given late in life to mice, it can actually reverse pre-existing age-related heart dysfunction. So having improvements in both diastolic and systolic heart function. Similarly, when started later or midlife, just a brief exposure to rapamycin treatment at relatively high doses can increase physical performance. So it can increase running distance and rotarod, which is another type of kind of, I guess, activity measure, if you will. And also rapamycin has been shown to actually pass through the blood-brain barrier. And so in a handful of different AD models, but also in just normative aging models, rapamycin is actually shown to improve cognitive function. And this is thought to actually be through improvements in cerebral blood flow. And cerebral blood flow is considered to be one of these kind of biomarkers for risk of Alzheimer's and dementia. And then last, both in mice and in humans, as Dudley mentioned earlier, short-term everolimus treatment, so about 6 weeks of everolimus, can actually improve the efficacy of the influenza flu vaccine in older adults. And so I think those are all really critical findings that suggest that rapamycin not only can extend longevity in mice and different preclinical models, but there's also a lot of data to suggest it can actually improve the health span or some of these key physiological outcomes. So Dudley, if you want to hit it again. Obviously, there's risk of adverse events, and I think Dudley has already discussed these, but these are all related to chronic, long-term dosing regimens that increase the risk of these different adverse side effects. Next slide. And so, you've seen a similar model that Dudley has mentioned previously, where when you give rapamycin acutely, you can inhibit mTORC1, and even when he discussed his genetic approaches where you can disrupt the mTORC signaling pathway, these all seem to have beneficial impacts on health span and lifespan in multiple models. And then, the chronic high dosing seems to be inhibiting mTORC2 signaling and leading to these adverse events. And so you can hit it again, Dudley. One of the things that we've really identified then through these series of really amazing preclinical work that Dudley's done and others have done is that there seems to be this need to preferentially inhibit mTORC1 signaling rather than mTORC2 to really capitalize on the health span extension while trying to minimize these adverse side effects. And so we want to think about trying to translate these data into human clinical trials and even perhaps just in mainstream, these are, I think, important aspects that we need to address. And go ahead, Dudley. And so kind of one study, we've talked about this Joan Manick study a couple of times, but I just wanted to kind of review some of their data because we're kind of using their kind of their prescription, if you will, for our upcoming study. But this is 6 weeks of daily, so 0.5 milligrams per day, or weekly 5 milligrams per week of everolimus. And then they showed is that it improved the influenza vaccine efficacy. And their primary endpoint here is that it needed to increase the efficacy by 1.2-fold in 2 of the 3 vaccine strains tested. And what these data show on the left is that this is full change in the titer response for the flu vaccine, and the two doses that I mentioned, the 0.5 milligram daily, the 5 milligram weekly, and they also did a higher dose, 20 milligrams per week. The first 2 are the 2 different treatments that actually met their primary endpoints of increasing over 1.2-fold relative to placebo for 2 different vaccine strains. So you can go ahead and hit it a couple of times. They also modeled that daily and weekly everolimus attenuates mTORC1 signaling, which is important to consider. And then when we look at the AEs, or the adverse events for the daily and the weekly low dose everolimus versus the placebo, there was really no difference in any serious adverse events. And the most common adverse events were transient mild mouth ulcers. So this was in about 11% of the everolimus treated group versus 2% for the placebo. And then there's also an increase in total cholesterol, or LDL, which is about 7% in the treatment group versus the placebo. So collectively, when we look at these data, they would suggest that low daily or weekly intermittent doses of everolimus appear to be safe, tolerable. They hit the molecular endpoint of mTORC1 signaling, and they improve vaccine efficacy in older adults. And so we wanted to extend these findings in our clinical trial. And so if we want to hit the next slide. The objectives of our trial is really to determine if everolimus can safely promote, I would say, multiple indices of physiological aging in humans, or health span. And then we also want to determine if everolimus selectively inhibits mTORC1 signaling and modifies hallmarks of aging in blood and skeletal muscle. And so we are going to be doing a longer term study, about 24 weeks, which we'll talk about next. But our hypothesis here is that we believe that everolimus will selectively inhibit mTORC1 signaling and improve metabolic, cardiac, cognitive, physical, and immune function in older insulin-resistant adults who are about 55 to 80 years old, and they are free of disease. And I'll expand on kind of some of the rationale for these objectives and hypotheses in the next slide. And so here's our overview. So this is a phase 2A randomized double-blinded clinical trial. And so, as I mentioned, we're going to be testing 24 weeks of everolimus versus a corresponding placebo. We are going to use the same kind of regimen as the Joan Manick study, since it had some promising impacts on immune and influenza vaccine efficacy. So we'll be giving them participants daily 0.5 milligram dose of everolimus, or a weekly 5 milligram dose. And our primary outcome is going to be measuring insulin sensitivity through an oral glucose tolerance test. But we also will have individuals wear continuous glucose monitors before, during, and after, towards the end of the intervention. We'll be measuring cardiac function through echocardiogram, since there's been some promising data in both mice and dogs. We'll be looking at physical outcomes, physical function outcomes through VO2 max testing, which is arguably one of the best prognostic markers of health span and morbidity in humans. We'll also measure skeletal muscle strength and power. Our cognitive outcomes are going to be using a really, I would say, an innovative 4D MRI of the brain to measure cerebral blood flow in both the macro and micro vessels, as well as doing some cognitive functions to evaluate memory and learning. And then last, we're using a similar protocol as the Manick study, where 2 weeks after they finish their last treatment dose, we'll be giving the influenza vaccine and testing their influenza vaccine response. In addition to that, we're going to be completing a multi-omics approach to try to fully investigate the mechanisms of aging by which mTORC1 can hopefully improve these fundamental biological hallmarks of aging. So that includes metabolomics, lipidomics, transcriptomics, proteomics. We will be doing DNA methylation, so we'll be able to look at a lot of these methylation clocks. We'll look at our immune cells, our mitochondrial bioenergetics, and then through recent funding through the impetus longevity mechanism, we'll be looking at the senescence-associated secretory phenotype. And so these are all really considered kind of biomarkers or hallmarks of biological aging. We're going to be recruiting older insulin-resistant adults. So we have 72 individuals in total. So there will be 24 that will complete it in each of our group, whether it's the low daily, the intermittent weekly everolimus, or the corresponding placebo. And I'll show you those groups in a bit. The reason that we are picking older insulin-resistant adults is when looking at this age bracket, there's a large proportion of the United States that are insulin resistant. Both age and insulin resistance are characterized by having hyperactive mTORC1 signaling in many tissues. And when we look at insulin resistance, well, we know that age is a risk factor for nearly every chronic condition, but insulin resistance further elevates that risk, especially for type 2 diabetes, cardiovascular disease, frailty, dementia, and immunosenescence. And importantly, these are all have been delayed by mTORC1 inhibition in model systems. And you can kind of see how our outcomes on the bottom left are very much related to the risk of these age-related conditions, right? So they're definitely clinically driven based off of some of the prior data. I think another unique aspect of our study is that we're going to recruit a young healthy group. So this is, your 18 to 35-year-old. They're not going to do any treatment, but they will complete all of these baseline evaluations. And so the goal here is that we want to make sure that if everolimus is changing any of our variables or outcomes, that they're changing them to resemble more of a younger, healthy phenotype rather than the opposite direction. So we kind of almost can look at them as a positive control group. And actually, if you want to go back real quick, Dudley, I forgot to mention that, these biological markers that we're looking at, we're going to be collecting blood, urine, saliva, and skeletal muscle biopsies. So we're really going to be able to look at some of these fundamental mechanisms of aging in a variety of different tissues that we'll obtain from our participants. This is just another way of looking at the study design. I don't think we need to spend too much time here, but we can go on to the next one. And so what I think we're doing, a really interesting approach to this study, and we're using what's called a double dummy design. And so what this means is that every single participant in the study is going to receive 1 capsule per day and 1 capsule per week. So they're going to basically be given 2 bottles, one that they take daily and one that they take weekly. So for the placebo group, this is placebo for both bottle A and bottle B. But for our daily everolimus, they'll be giving their daily bottle will be the everolimus, and the weekly bottle will be placebo. And then this is the opposite for our weekly everolimus, where the 5 milligram weekly will be given a placebo every day, but then the 5 milligram of everolimus weekly. So this is really, I think, a sophisticated approach because it not only blinds us to the active study drug, but also versus placebo, but also the timing. And so we won't know which groups will have the drug on a weekly or daily basis. And we're recruiting more people to enroll in the study. That's why you see the different numbers here. That just accounts for anyone that may drop out as the study progresses. This is double-blinded. We will not know, and the participants will not know what they're on. And we are trying to achieve an equal distribution of both men and women. Historically, in our studies, so my kind of background is trying to run these clinical trials, especially geared towards kind of these geroscience guided trials. we tend to have a lot more women participate in our studies, and so we have to work really hard to try to get men incorporated. So that's why that asterisk is there. And I think that might be it, if I recall. Yeah.
All right.
So I think, yeah, Dudley and I would love to entertain any questions, and yeah, I guess we'll go from there.
Absolutely. Well, thank you very much to both Dudley and Adam for your fascinating presentations regarding your research, and certainly the Everlast study that you outlined seems to be quite promising. We have a number of questions already from our panelists and our audience. I would like to start with Dr. Bill Andrews to see if he has any questions or comments.
Yeah, thank you. Well, first, this is not exactly my mTOR and rapamycin is not my main field, but I'm very, very interested in this. I've been following the science for quite some time. And one of the big turning points for me was Dudley's Motor paper. That paper, I was very excited about that paper. I read it about 6-7 months ago, and it's been something that's gotten me a big follower of everything going on with rapamycin. I am curious, since metformin is also an mTORC1 inhibitor, is there any advantage to taking both metformin and rapamycin?
Well, Adam's our metformin expert, so I'll let him take that.
Yeah, that's a great question. So the ITP, which is in mice, so as Dudley alluded to, showed some of their data. When they combine rapamycin and metformin, there seems to be, compared to historical cohorts of mice, there seems to be a more potent effect at extending lifespan, whereas metformin alone had no statistical advantage. Now, as far as humans, there's really no data that's publicly available to suggest whether or not metformin might have an advantage when added to rapamycin.
Okay. And you know, there is a clinical study underway that's been going on a few years to look at metformin and extending lifespan in humans already.
Well, that trial is not yet started to my knowledge.
Okay.
That would be the TAME trial, correct?
Yeah.
That would be the TAME. Yeah, the TAME trial is a pending trial that will look at the impact of metformin at improving health span. It's a 14-site clinical trial. We're recruiting over 3,000 individuals. It could be a really exciting trial, but there's been actually I don't understand. I don't know what's going on with that trial, but I don't believe it's actually started yet.
That is news to me. I thought they'd already started the TAME-2 trial. Because they got data from TAME-1. Okay, so my other question is, now the thing that I've always heard about everolimus is that it's an mTORC2 inhibitor. It inhibits mTORC2 better than rapamycin. And so is that not true, or why? I had to walk out because I'm supposed to speak at another conference. It's actually a celebration of life for a colleague of mine, a scientist that passed away recently. But but they've delayed my talk. You might have said something about mTORC2 and everolimus, or did you say anything about why you're doing everolimus instead of rapamycin in your study?
No, we didn't really talk about that extensively. In some mouse experiments we did, we saw that dosing mice with equal quantities of either rapamycin or the analog everolimus, both of those had immunological side effects. Both of them weren't great in inhibiting mTOR complex 1, but everolimus had less side effects related to glucose tolerance, so less metabolic side effects. And while I am not by any means an everolimus expert, my understanding overall is that it seems like everolimus is cleared more quickly. And so you can imagine that you get more of a peak of mTOR complex 1 inhibition and sort of clear it out before there's as much mTOR complex 2 inhibition. I don't think that there's any data suggesting that everolimus is better than rapamycin at mTOR2 inhibition.
Oh, okay. So I thought I read that somewhere. Okay, so
And Dudley, does your mouse data would suggest that actually everolimus has less of an impact at inhibiting the mTOR2 signaling pathway? Is that correct?
That's correct. Probably related to PK rather than enzymatic specificity.
okay, if you don't mind, I'm going to go back and find out where I heard that, and then get your comments on it after this meeting. My last question was related to, it's not a rapalog, but it's a kind of rapamycin packaged in a nanoparticle, and it's called SVP Rapa, who recently got the trade name of IMMTOR, IMMTOR. And it's something that I've been watching only because it has had significant effects on reducing host versus graft disease, and also in mice at least, and a significant effect in making it so that animals, mice, maybe even humans, don't have an immune response against gene therapies like adeno-associated virus. And so I've been watching this, but at the same time, I've been curious, why does it have these, not have these effects, like why it almost eliminates host versus graft disease? Is it targeting different pathways than just regular rapamycin? Is the nanoparticle taking it somewhere else that rapamycin goes that causes some of the side effects? And are you aware of any studies at all that have looked at mTOR on its ability to affect the same pathways that rapamycin does?
I'm not familiar per se with IMMTOR. Maybe Adam would like to talk about it. But, overall, that elimination of graft versus host disease is exactly the, major indication for rapamycin and everolimus and temsirolimus. It's designed as an immunosuppressant for organ transplantation. That's its sort of primary use. Whether this might have slightly better effects or be targeted more specifically, I don't know. But, sort of in general, it's a very similar indication, it sounds like.
So somebody just put up the link to one of the papers by Takashi Kishimoto. I've talked with him a lot about this because of my interest in the gene therapies. So I don't know who put that up there, but
I did.
It sounds like maybe there's some sort of antibodies that are generated to rapamycin in some individuals, just from the title of the paper.
Yeah, maybe. I don't know. Okay, so good. I'm just a big fan of this research. I'm following it closely, and so thank you very much. That's all my questions.
Yes. Thank you very much to Bill for those questions, and to Dudley and Adam for your responses. I have one question, and this is just a matter of curiosity before we go to the other panelists' questions. So I noticed in one of the tables early on that Dudley presented, it seems that rapamycin extended the female mouse lifespan more so than the male mouse lifespan, especially earlier on in the lives of those cohorts. So the curve was pushed further out for the female mice. And have the reasons for this differential effect been studied?
There's definitely interest in understanding why that is. What I can tell you is that across all interventions in the sort of PI3K signaling all the way downstream to S6K, which is downstream of mTOR complex 1, every genetic model that's been made in a mouse, be it inhibition of S6K1, inhibition partial or inhibition of mTOR signaling, inhibition of insulin receptor substrate signaling, insulin receptor signaling, PI3K signaling, AKT signaling, all of these interventions, they all work better in females than males, unless you increase dosing for rapamycin, and you can sort of get them to work equally well. So whether that's a dosing response or whether females are just more primed to having a better response to rapamycin for reasons that we don't know, it seems like this is a pretty conserved response. And I'll say that the NIA's intervention testing program has found other drugs that work better in males. So there might be interesting combinations therapies in the future too.
Yes, thank you very much for that response. And now let us go to Jason Geringer. Jason, I know you have a few questions.
I was wondering if you could talk a little more about the hyperlipidemia issues and how rapamycin has effects on that, and is how much of an issue would somebody with high cholesterol, have. And I was wondering if you could maybe just explain the difference between acute and chronic when it comes to like dosages and that kind of stuff. I mean, most people probably know what it means when it comes to pain, but and then also I wanted to know, this study you're talking about, this was a study you guys are going to do and it's coming up and pretty soon then, or what's up?
I'll do lipids, and then maybe we can jump to Adam, because there's a lot of questions here. So just to sort of do lipids first, we don't really know what lipids, what the consequence of hyperlipidemia induced by rapamycin is. There's one mouse study that suggests that rapamycin is good for cardiovascular disease in a mouse. Just one study that I'm aware of, so it's not been explored to extensively in any event. And more importantly, mice have a lot of dissimilarities between humans. So mice actually almost never get atherosclerosis normally, even if they're on terrible diets. And of course, humans do. So exactly what the consequences are, we don't know. And one reason we don't know is that people who take rapamycin for organ transplantation in particular, there's a zillion immunosuppressants now. And so if people get hyperlipidemia on rapamycin, typically their doctor is just going to switch them to a different immunosuppressant. So we don't know what the long-term consequences might be. I'll let Adam take all the study-related questions and so on.
Yeah, so your question, Jason, we are about to start our study. So we're trying to get off the ground here in the first quarter of 2023.
Did you want to take chronic versus acute?
Yeah, so chronic versus acute. So we have, so we're I guess technically we're incorporating both. So we have a really low daily dose and then a intermittent weekly dose. And so we think that we've found a dosing scheme that's low enough to avoid some of these off-target side effects, as well as trying to capitalize on Dudley and others' data to use this intermittently weekly dose to, again, help minimize the impact on mTORC2 signaling and adverse events.
Adam, do you want to talk about what a sort of chronic dose for immunosuppression or cancer might be in people?
Great point. Yeah. So there's a wide range, but individuals taking this for the FDA-approved treatment indication, it can be anywhere from 1.5 to up to 10 milligrams per day of everolimus. So you can kind of appreciate that we're really at a anywhere from a 30% to even a much smaller amount than that for our dosing scheme. So it's a very low amount. So a lot of we hope, and the whole idea behind this is that we expect these low dosing schemes to really minimize the adverse events. And that's similar to what Joe Manick has found too.
So that would be the acute, those lower dosage, that's an acute. Is that what you guys mean here? Or
the lower dose is meaning we're going to give it daily. So every single day for 24 weeks. So that would be a chronic, dosing scheme, chronic daily dosing scheme. But we think that it's so low based off of the human and animal data that we will hopefully minimize many of the adverse side effects that we see in patient populations with a higher dosing scheme. And then
so you guys got a different kind of rapamycin. I noticed you mentioned rapamycin, and then there was something else, and then a D001 or something. Are those like the same thing, but just changed a little to get rid of some of the side effects, or what? Can you explain about those?
Yes, I I think Jason was referring to DL001.
So DL001 is an investigational mTOR inhibitor that we published on a few years ago, made by a company. Everolimus is a compound that I believe was made by Novartis. It has a small chemical modification that probably has a variety of different purposes. Adam, I don't know if you want to talk about what those purposes are, but several different similar chemical modifications have been patented by different drug companies, and everolimus and sirolimus are the ones that are orally available for humans. Sirolimus is just another name for rapamycin.
Interesting.
Cool.
So rapamycin and sirolimus are synonymous. That's good to know. And Bill, you were actually referencing another paper that I can post the link to shortly, which is entitled Everolimus and Sirolimus in Transplantation Related but Different. Could you perhaps speak a little bit as to what you saw in that paper?
Well, I told Dudley I would send that paper later, but I found it really quick. It's the paper that I thought said that everolimus was better at interacting or inhibiting mTORC2, but I'm actually thinking that I might be misreading that paper. But that was what led me to believe. Let me bring up that abstract right now. Look at it. Is it just interacting or is it inhibiting?
I mean, that it does appear to be what it says, but to my knowledge, that is not true, at least in cell culture. Whether it's true in an organismal, in this particular organism, that seems to be, but we can certainly investigate. Certainly seems to have less mTOR complex 2 mediated side effects in the mice that we've looked at.
Yeah, I'm very familiar with irreproducible studies, so good. I'd be interested in what you.
So your study's using everolimus and not rapamycin, then?
Adams.
Yes, that's correct. And so part of that is due to some of the pharmacokinetics, as Dudley kind of alluded to earlier, it gets cleared faster. And so we think that because of that, we can hopefully more selectively inhibit mTORC1 signaling and really capture some of those benefits and really minimize our ability to inhibit mTORC complex 2 and the side effects.
Yes, thank you very much. And now we have a related question from our friend Daniel Tweed, who wonders, are other anti-organ rejection drugs generally being studied for possible lifespan or health span effects? So rapamycin or sirolimus and everolimus have been studied, and then DL001, Dudley discussed a bit in regard to the studies about that. Are you aware of any other anti-organ rejection medicines that might have those kinds of effects and are being studied for those effects?
Well, that's an interesting one. I don't think I've heard of them. Adam?
No, that's a great question. I actually don't know what some other common transplant drugs are, but it seems like it could be
I mean, tacrolimus is the one that's sort of often dosed with rapamycin or compared to.
Right.
And it has a vaguely similar mechanism of action with regard to calcineurin. So might be something interesting to look at.
Yeah.
Yes, thank you for that response. And another question from Daniel is, has rapamycin been studied in comparison with caloric restriction for longevity effects?
We'd be delighted for someone, if they're interested in funding some of our studies in that regard. I can say that the various funding agencies we've applied to have not been particularly interested in supporting that question. I think one question, is certainly, how those might interact from a beneficial standpoint. But Arlen Richardson and Viviana Perez and a few others have done a bunch of short-term studies with rapamycin and calorie restriction. And the surprising finding from their studies is that from a molecular standpoint, it seems that they have very different effects in the liver and the white adipose tissue on the metabolites that are in the blood. So we've been really interested in the idea of trying to expand that into a larger study where we could actually sort of compare those from a lifespan standpoint and a health span standpoint and test whether they're additive. So that's something that definitely anyone who's interested, feel free to reach out.
Yes, indeed. Well, I'm glad that you're interested in exploring this question. I hope that you do get the funding. I know sometimes the funding bodies can be a bit difficult to persuade for various reasons, and we can discuss those reasons perhaps later in the session, but I think this concept is quite intriguing to study. Here's another question from Chasing Bob. He wonders, with regard to the Everlast study, will you be testing the VO2 max of the older group using a treadmill test?
Great question. So we are actually going to be using a cycle ergometer or stationary bicycle. So same concept, just a different mode. Generally speaking, for some of the older adults, it's a little bit easier to get on and off the treadmill, and then we don't have to worry about them flying off the back of the treadmill either.
Excellent. Well, that does seem like a sensible approach. And I'm a little bit familiar with the treadmill tests myself, so I can definitely see where you're coming from. Now I want to go to Ben Ballweg to see what questions he has.
I just have a quickie about mTOR, et cetera. You did mention about Richter getting knocked out. I wonder what happens with Raptor getting knocked out.
Oh, Raptor is a very essential gene. You do not want to knock out Raptor. If you delete it in virtually any tissue, you get sort of very strong anti-proliferative effects. If you successfully keep it knocked out, very bad things happen for health. A lot of the time you get some escapers though, and it sort of reproliferates. So in fact, that's something that's sort of an interesting tangent, which is that a lot of people have studied mTOR kinase inhibitors, which target the kinase activity of mTOR complex 1 rather than rapamycin. And it's an interesting question whether those would be better or worse from rapamycin from an anti-aging perspective, because mTOR complex 1 being inhibited by rapamycin is a little bit of a simplification. A lot of the activities of mTOR complex 1 are inhibited by rapamycin, but some activities are not. And so if you knock out Raptor or you treat with a kinase inhibitor, you're actually inhibiting a different set of downstream processes. And that might be beneficial from a cancer drug perspective. That's why a lot of people have been interested in it. But it might be beneficial or negative from a lifespan perspective. And I think that's something we don't know a lot about.
Very interesting. Thank you very much for that response. And I have a question that follows up a bit on Bill's question. So he was wondering about investigating metformin in conjunction with rapamycin to see if there are any additive benefits to lifespan extension. And it seems to me that there is a bit of a complementarity there, because metformin is an anti-diabetic drug, so it helps lower blood glucose levels, and rapamycin has the risk of essentially increasing the glucose generation. So do you think there's a realistic possibility that the two would offset each other in a certain way to keep the glucose production at reasonable levels?
Yeah, I believe there's data from Adam Salmon's lab that has shown that in mice. And I believe it is in one sex, and then it's escaping me which sex. Dudley, do you remember?
I don't, but you're right. It's only one sex.
It's in one sex. And so, yeah, it can help protect some of those adverse effects on the glucose metabolism. I guess the kind of question I always have is, do you want to take something that causes an adverse event and then take another drug to help cure that adverse event? So it kind of comes back to me as maybe we should really be focusing on what are the appropriate dosing regimens that can safely improve health span and lifespan and go from there, instead of trying to take a really high dose of rapamycin and then take another drug to help offset some of those adverse effects.
Yes, that makes sense. That is also a sensible approach to prevent the adverse effect from happening in the first place. Now, let us go to, yes, go ahead, Jason.
Would you say that might be what's going on with the metformin and the rapamycin, that the metformin is sort of just like cleaning up maybe some of what the rapamycin is accidentally doing, or I'm just wondering why they work together, and I think maybe you might have answered my question by saying that sometimes, another drug will help to clean up the side effects or something. I'm just wondering if you think that's what's going on here with it.
Well, there are some people who take just metformin or just rapamycin, and they at least hope for life-extending effects. Of course, for metformin, we would want something like the TAME trial to happen to really be certain that there are significant life-extending effects for humans. But even people who take them in isolation think that there's some longevity benefit. And there have been mouse studies suggesting that there's a longevity benefit of each one of them in isolation. The question is, can metformin help offset some of the adverse effects of rapamycin? And that's what the thinking of taking them in combination would be. So now I would like to...
I'd just like to sort of jump in there to add something, right? So one thing that's very clear is that metformin is good for people with diabetes, right? Not only does it protect them from various, complications related to the disease, it's been known for 10 plus years that it extends their lifespan. And so if you have diabetes, metformin is very clearly beneficial. And so I think, if rapamycin gives you diabetes, then it makes sense that, metformin on top of that would sort of offset some of those effects. But the flip side that is like polypharmacy, right? Taking more and more medications in mice and in humans is associated with frailty and poor health. And, we don't know causation exactly, because in people, you're taking more drugs if you have more diseases. But at least in mice, just giving mice more drugs, on its own, it tends to promote frailty. And so, just taking more and more drugs might not be the best approach either.
Yes, indeed. There's a comment in our chat from Matt Alex along these lines. He writes, as Brian Kennedy pointed at, it's better to focus on some few supplements which are proven effective than to multiply to a dozen of those substances, as the interactions can be deleterious. Now, are you aware of any possible adverse metformin rapamycin interactions that people should watch out for?
I mean, I'm not, but I will certainly say that I've talked to a number of people now who are taking rapamycin for its potential longevity benefits. We're just spinning up a observational clinical trial where we're going to be advertising in a month or two in the new year to try and recruit people to send in their blood samples if they're taking rapamycin, so we can study them. But I have talked to some people who have reported that there were negative side effects, and that they believed that they might be due to some of the other medications they were taking. Not in this particular case metformin, but other various supplements. Whether that's true or not, we don't know, right? Because it's all of these individual anecdotes are just individual pieces of data, and we don't know how that would be put together in a broader trial.
Yes, that makes sense, but it is interesting that the question is beginning to be explored. Now let us see if Art Ramon has any questions or comments.
Yes. Thinking how mammalian evolution, why it would sort of increase lifespan on an organism. So like if you have insufficient calories, Okay, you don't want to have offspring if you don't have the calories to grow offspring. So that's one reason I think evolution would do that to mammals, is extend their lifespan until there is a time there is more sufficient calories. And also, if especially a mammal, if you don't have a mating partner, I mean, you're not going to be able to reproduce. So that also evolution would sort of build in something to extend lifespan if you aren't able to find a partner. So that might be the reason why it works more with the female mouse in those prime early reproductive years. You would extend that. Nature would do that through evolution. So is it possible that the studies could be skewed if the sexes are separated?
It's an interesting question. You know, mouse studies, typically the mice are separated, but they can smell each other. And so a lot of those signals might be able to still be transmitted from cage to cage. So there might be some interesting possibilities in terms of promoting greater separation. But I will say rapamycin still works in females and works better than males when started at 20 months, which is post reproductive period for female mice. So even works just in that post reproductive phase.
Interesting. And I don't think it's quite that reproduction shortens the lifespan of the organism. It's more that organisms that have had to endure periods of prolonged resource scarcity evolve longer lifespans in order to be able to reproduce at some period when there are abundant resources. But also, there is the observation, and Aubrey de Grey and a few others have mentioned it, that essentially evolution stops being concerned with your survival once you've reproduced and perhaps raised your offspring. There is also the grandparent hypothesis, so there might be some sort of adaptive function to having some older individuals around, but essentially the mechanisms that protect one until one has reached reproductive age are no longer operative, and that's one reason why biological aging happens. I don't really see whether or not one has reproduced as determining one's individual lifespan or lengthening or shortening it. Would you agree with that observation?
I mean, I'm not aware of any data directly on that standpoint. Certainly, though, particularly from a female perspective, reproduction is very stressful and a mortality risk. Adam?
Yeah, I'm not sure if I can comment on that.
All right. Well, we have another question from our friend John H., who wonders, are we getting close to the time when I can get my 25-year-old body back? He writes, I miss it more and more as the days go by. And I would echo that, John. I think perhaps we all would. But what would you say in response to your question? What is your view as to the time frame for achieving that kind of biological age reversal?
Well, Adam may have different thoughts, but I mean, I think most of the interventions that we're looking at seem to be interventions that slow the rate of aging rather than reverse aging. Even though, as Adam pointed out, rapamycin might be able to sort of rejuvenate the aging heart to some degree based on mouse studies, and that could be very beneficial. I did read a tidbit recently that people only begin to think of themselves as old at about 47 now in the United States. So, anything under that might still people might still be thinking that they feel young.
Interesting. And Adam, Dudley suggested you might have a bit of a different perspective. What would you like to add in regard to that question?
I don't know if I have much to add. I think, yeah, Dudley's right. So far, it's been more of the slowing or preventing of some of these age-related conditions. There's been some data in mice that have used actually some rapamycin nano or microparticles. This is still preliminary. I don't think it's published yet, but when injecting these particles into the knee joint of the mouse, they can actually help rejuvenate some of the age-related and injury-related arthritis that can occur. So perhaps there's promise for different specific tissues, but I think right now, I think we could probably just say that it would slow or prevent some of these age-related conditions.
Mm-hmm. All right. Well, thank you for that response. And our friend Mike Lazine notes that he is 47 and he does not consider himself old at all. So perhaps he is not like the statistical mean American in terms of his outlook on aging. So I wonder also, since we touched on this issue with regard to funding and how easy or how difficult it is to fund particular kinds of studies given the funding bodies that are out there. Could you perhaps comment on some of the considerations and some of the challenges involved in that?
Sure. I mean, sort of overall, the funding rate for most grant proposals is on the order of about 15 to 20%. And of course, that means that a lot of grant proposals need to be submitted in order to, get a funded proposal in. And, some ideas are more topical at certain times than others. And so as a result, it may be more relevant, easier to get funding at certain times than others. I think there's a lot of interest right now in starting to translate some of these interventions to aging. So that's beneficial. And I think one of the reasons that, Adam's been successful in getting some funding for human aging trials, and hopefully we'll be getting some additional funding in the future. But, I think it's a great time overall because there's a lot of interest in the aging field right now and trying to think about interventions. But there are also a lot of great ideas out there too. So, I think there's always probably going to be more good ideas and good teams out there than, there can be funding for. So it doesn't surprise me that, difficult to get funding for individual ideas here and there. But it is sometimes frustrating when, we have good questions that have sort of been unanswered for a long time and that we'd like to be able to get funded.
Yes, indeed. So,
Yeah.
Adam, go ahead.
Yeah, I think another thing to consider there too is, the cost of doing cutting edge research has skyrocketed. Much of the other aspects of the country, right? As inflation has really gone through the roof, and so what you can get done on a standard modular R01, which is a common funding mechanism, what you can get accomplished is actually a lot less now. And so those budgets haven't really changed in, I think, the last 20 plus years, where obviously the cost of doing research has changed dramatically. So I think that's a big hurdle. And then, the other aspect too is when you look at the TAME trial that was proposed and it should be starting soon, hopefully, that's like a $50 million budget. It's a big study and it requires a lot of funding. And so hopefully, through some of our initial studies and some other studies that our collaborators or colleagues in the field are running, hopefully we can get to the point where we can, start having these large scale rapamycin or rapalog trials where we can actually make some really big, I would say, differences in the aging field. But it just takes time and it takes these kind of like stepwise progressions to get to that point.
Yes, this is quite interesting. Jason, go ahead.
Where's the problem? Where's all the money going? How come it's so expensive? And how can we make it easy? And what would be, where's the problems and where's the ideal way we could do this, do you think? If you had your, magic wand and could make the studies how you want, and, how could we fix it, do you think?
Yeah, I think that's a $1 million question right there. We have to cover for, we have to pay people to do the work. We have to, pay our volunteers to participate in the study. the genetic and kind of really sophisticated techniques, they're not necessarily cheap. And so all these things start stacking up. So it can get
So you
expensive really
pay
quickly.
you pay your participants? Like if I was going to be a guy that ended up being in the placebo group, you got to pay them?
Yeah, so we compensate people for their time and their efforts and travel, et cetera. And so, for our 24-week intervention, someone can get paid up to $2,000 to complete all aspects of the study. And if they drop out or if they don't complete part of the study, they get a prorated amount.
Okay, so what are all of the rest of the things of the study? Do you actually have to go somewhere? You can't do it remotely, right?
Unfortunately, for this study, we're a single site. So this is still kind of I would say, smaller in scale from that perspective. It's 72 participants in one site, and we have a lot of, I think, kind of highly sophisticated techniques that our university permits us to do and allows us to do. But I think when we start thinking about how do we scale up to more of a phase 3 clinical trial, then hopefully we can get more access for people across the country. So again, we could have sites all over the country doing some sort of rapamycin or rapamycin analog trial.
So they're going to live there? They're going to go there and move in and live there for how long?
Well, for us, this would mainly be people who live in the area. So generally speaking, individuals that live within an hour of Madison, Wisconsin, would be willing to participate from our experience.
Just as a sort of building on that a little bit, right? Everything costs money. So we're doing a much smaller scale study that's observational, looking at people who are already taking rapamycin and paying for it out of their own pocket. There's, all across the United States. And so we have some impetus funding, and we're going to launch this trial. And basically, we're going to have somebody here in Wisconsin who's going to work with a company that has phlebotomists across the United States. And phlebotomists are going to go to people's houses and draw their blood and send that sample back to us. And so, you can think every individual step costs money, right? You got to pay the person who draws the blood. You got to pay for their gas money. You got to pay for the tube they use to draw the blood into. You got to pay for the mailing of the tube back here. Once it gets here, we have to have a technician who separates out the plasma and the whole blood. And just then, right, then the clinical tests can start. And then we can take the data and send it for high throughput sequencing and looking at metabolomics and lipidomics. But then we're not done, right? Then we got raw data. And then we need to have people who can analyze that, who know how to do all the complicated bioinformatics techniques. And then, overall, so that study is actually going to be very sort of focused, short term. We're going to have about 100 people in that. As well as about 100 controls probably. But overall, right, every one of those things is true for Adam's trial too, but it gets even more complicated because they got to come in, they got to get their blood drawn. Many clinical labs have to be taken at multiple time points. If you take a tissue sample, you got to sterilize the instruments, right? And somebody's got to pay to sterilize the instruments before you take a muscle biopsy. So everything.
Yeah, a lot of that money, I must say, goes to inherently making sure that the study itself is going to be accurate. If you're just crowdsourcing, Joe Schmo's data, that, you know what I mean? Like you just said, if the procedure calls for you got to sterilize the instrument. Well, you got to make sure that happens to keep the data sound. And so you have to be able to prove that. So to do that, you can't really ask a volunteer to prove that. So you probably got to pay them. And so, but I imagine there might be some maybe shortcuts you can get in there. For instance, I do blood work already. There's probably some data you could get just from volunteers, but again, that's
I think that's a good point, Jason. Actually,
data is you're probably getting the way you're doing it.
So I think that's a good point, Jason. So yeah, you can do that. And I think there are folks that are trying to do that. Is like take people who have been taking rapamycin for X amount of years, kind of like what Dudley's doing, and then you can get some of the biological data. You can also get some of their their perceptions of how they're feeling, et cetera. But what's kind of lacking from a scientific value is the placebo effect. We're doing a randomized, double-blinded controlled trial, and so there's a large placebo effect when you're talking about supplements, drugs, et cetera. And so you really need to account for that. And so that's one of the values of having these kind of highly controlled studies. They might be expensive, but from a scientific perspective, there's, in my opinion, probably no better way to perform a study.
Yeah, and I would think
just
it's
sort of
inherently.
I think, I don't recall exactly what I've heard Joan Manick say on the subject. Maybe you do, Adam. But, she did this very large study, several large studies involving about 1,600 people in total taking various drugs or placebos. And everyone who took the placebo said that, they felt better. It's like I think about half of the people said that they felt better. So they all thought they were on rapamycin, but they weren't. They were on the placebo. So there was a huge placebo effect in these studies.
Right. And I imagine it's probably inherently difficult to get somebody to volunteer and then not know that they're on a placebo. I mean, in a way, when they're self-reporting data, I don't know how that would work. You can't trick yourself. I don't know. Hard to make blind, I guess. Yeah. Go ahead.
Yes. Ben, you had a question or comment that could dovetail with this discussion.
I'm of the impression that you have both been working, I can't say exclusively in aging, but I wanted to know specifically challenges of researching aging versus not researching aging, or researching not aging.
Aging research takes longer. And when we do lifespan studies in mouse, right, it's going to take 4 or 5 years to sort of see what the effect of an intervention. So that's sort of a long, expensive study. You sort of have to have the right mindset to be patient. Even if you do a life intervention, it's still going to take a couple of years. And, for various other disease fields, that's a really long period of time. And of course, you can just imagine how patient Adam's going to have to be for his studies if he eventually starts looking at human lifespan on rapalogs.
Yes, and it seems based on your comments that there's more interest in research into aging now and perhaps more of a willingness by certain institutions to fund it, but any given dollar of funding will not go as far as it used to because of inflation and perhaps other kinds of constraints. So what do you think can overcome those kinds of limitations?
I think there's been a lot of interest in setting up some more core type of resources that are available to aging researchers. One sort of limit on doing age-related research in lots of fields is the availability of aged animals, and both some private funders as well as the NIH have been trying to expand the availability of aged rodent colonies to do experiments in aged animals so that we could see how they differ from younger animals and find things that are really specific to helping an animal that's already older. Because a lot of people, for cost reasons, do experiments only in young animals. And I think there's a growing awareness that that's not okay, or at least that doesn't necessarily yield results that are applicable to older individuals.
Right. Yes, especially when it comes to the prospect of reversing biological aging, starting with mice or other animals which are already older and seeing if particular interventions will have effects is especially important. And I have heard other scientists tell me that getting aged mice is sometimes a challenge and can be expensive as well. So it's interesting that getting an older mouse is more expensive than getting a younger mouse And presumably that is because the mice have to be kept in certain conditions and aged to a point where they're still healthy to a certain extent before the trial begins. And that takes time and that takes effort.
Yep. I mean, housing for mice is usually on a, a cost per day basis. And that makes sense, right? Because you have to pay people to take care of them based on how long they live. And so I think it makes sense that if you have to keep them alive for, a year and a half or 2 years before you start studying them, it's going to be a lot more expensive to keep them alive than if they only have to keep them alive a couple months. So, and everything associated with that, food and electricity and so on.
Right. And of course, one of the, go ahead.
I was going to say, maybe I'm changing subjects a little bit, but one, I think, aspect or challenge that we haven't talked about too is that as we age, there's an increase in the heterogeneity of the aging response, and so there's a lot of variability from animal to animal, person to person, and so to try to account for that, you need larger sample sizes, and so I think that variability is it's a beautiful thing because I think we can learn a lot from that heterogeneity, but it can be a big challenge too. And so I think that's another aspect that, from a biological standpoint, that's a challenge that we have when we work with aging models.
Mm-hmm. Yes. So by the time that they're aged, they're already a bit heterogeneous as well because of their life experiences prior to the start of the experiment. But of course, another aspect that sometimes frustrates me in some of the mouse studies is that the studies end at a particular chronological endpoint, and then the mice are euthanized, even though they could have lived longer and scientists could have discovered the true extent of the life extending impacts of a particular intervention. So what do you think could be done, or what do you think would need to change for more trials to enable the mice or other experimental animals to just live out to the conclusions of their natural lifespans so scientists can get even better data?
I guess I would say, a couple things. One is, right, most scientists will be thrilled to add additional groups for longevity onto their studies if, funding was available. So maybe more targeted, funding sources to do those lifespan studies on top of whatever disease indications might be studied. But I would also say that, phenotyping and taking care of mice as they get older is not necessarily for everyone. And certainly there's a decent variation between facilities as well in terms of what a natural lifespan would be. So it's not clear that necessarily that would be the solution, I guess I would say.
Interesting. I do know that the record-setting lifespan for mice was a little bit under 5 years, and this was done with genetically engineered Ames dwarf mice by Dr. Andrei Bartka, but that was in 2001, so it was 21 years ago now, and I think it would be great to have some study to try to break that 5-year age barrier for mice, because I think that would raise public awareness and certainly get more members of the public, more institutions interested in the study of aging and how far lifespan extension can be taken in certain animals, hopefully eventually in humans.
Definitely, I agree. Getting some super long-lived mice would definitely be very exciting.
Yes, indeed. So one of the aspects that we delve into in our virtual enlightenment salons is the cultural and societal aspects. So we here are all proponents of longevity and life extension and figuring out ways to extend healthy lifespans. Sometimes we have encountered skepticism regarding these aims in the broader culture. And I'm curious as to your observations of how those attitudes might have evolved over time. given that institutions are a bit more receptive now in terms of funding research on aging, do you think the culture has shifted in a more favorable direction as well? What are your observations on this?
Well, I think people are buying into the idea of healthy longevity. So I think there's probably multiple camps of people. There's some that are going to really be prefer to extend health span versus lifespan. Some that say that those things can't actually be uncoupled. But yeah, I guess I don't know, Dudley. What do you think?
I mean, I think that there's certainly a lot of buy-in for the idea of healthy aging, right? People don't. I think that is a sort of a bit of a change, because when I started getting involved in this field, people typically thought of extending lifespan as extending the unhealthy portion of life, and I think there's broader awareness now that extending lifespan might really be extending health span and getting to the end of our life in a sort of a strong, vigorous way. Certainly, though, I mean, I think there's a lot of skepticism both in the public and in scientists about people who are talking about turning back the clock. And there's certainly been some molecular biomarkers, say methylation clocks, that show rejuvenation in various species, maybe even in humans sometimes. Whether those will eventually turn out to be, clinically useful and wind up extending human lifespan in addition to human health span, I think remains to be seen.
And of course, it's interesting because sometimes one can make progress in one area of aging on one type of aging-related damage, but there could be other types of aging-related damage that still accumulate and cause life expectancy to be about the same. And this is why Aubrey de Grey, for instance, holds that a combination approach is needed to ameliorate all of the major types of aging-related damage at once, so that one could see meaningful lifespan extension and rejuvenation. But it's interesting, too, that so many people have historically held this fallacious view that life extension would mean the extension of the ill and frail period. It seems to me it would be quite difficult to extend that beyond current maximum lifespans today, because when people die, they die of some conditions. So a healthy person doesn't just drop dead all of a sudden. And Bill Andrews, who unfortunately had another commitment and had to depart from our session, likes to say that one cannot extend lifespan without also extending health span. So would you agree with that observation? And do you think, if that is an observation you agree with, that there could be some more effective ways to show that to the public, to convince people that this is indeed the case, that healthy lifespan extension is really the only lifespan extension that's possible beyond the limits that exist today?
I guess I would say that you've touched on something important, right? And that's, for the last, I don't know how long, 40, 50, 70 years, right? A lot of the interventions that we see are interventions that extend like the last 6 months of life in sort of an unhealthy life. And so I think that, maybe people have got the idea that we're talking about extending unhealthy lifespan from those medical interventions that, are sort of heroic, and, people wind up on respirators or, in a nursing home for their last few months of life. And so definitely, I think that, that we're much more interested in extending healthy aging. Certainly from a, animal standpoint, it does seem that, those can be uncoupled to a degree. And so I don't know that extending lifespan always involves extending the healthy portion of life. But certainly we want to find interventions that do, because no one's interested in extending the unhealthy lifespan.
All right. Well, I'm aware that Jason has a question as well. So Jason, please proceed.
Yeah, I got a weird question. It's kind of like a hypothetical, ethical question about science itself. And it comes up because we're talking about anti-aging, and LEV, longevity escape velocity. I mean, what happens if this study here was the one that, and then somebody was in the placebo group and then didn't make LEV because, he was in the placebo group. Is that not ethical? I don't know. Just a weird question, but have you ever had any problems with people being like, "Hey, I'll do a study, but I don't want to be in the placebo group. Can you hook me up?" Has that ever happened?
Well, I mean, we're double blinded, right? So we don't know, and they won't know either, right? So there's no way. In my experience, we have had people that have dropped out of studies when if we're doing like an exercise trial. And so we often will have a sedentary control group. And if they get randomized to the sedentary control group, they're no longer interested in being in the study because they want to be in the exercise training group. So we do see it from those kind of more lifestyle modification studies that, people who are in some sort of control that don't get access to whatever the lifestyle modification is, then yes, they will drop out of the study.
There have been studies I'm aware of where for certain medicines that have shown to be extremely efficacious in a life-saving way, the placebo group, once this was apparent, was reassigned to the group that actually received the medication because of ethical considerations, because the scientists determined that it's more important to try to save their lives.
Yeah. And there's also study designs where you get standard of care, right? So you get standard of care that's, good. And then they're testing whether or not it's a superior, is this like a superiority trial? Is this a better medication or better intervention to further enhance the benefits that we see with standard of care?
We can only imagine that if, these studies turn out to be successful in the long term, right? Future longevity studies might use rapamycin or metformin or who knows as the standard of care.
I mean, that argument has been made before as, so as far as data in humans so far, I mean, exercise or diet could probably be argued as one of the, most important lifestyle modifications that one can make. And so the question becomes then, do we actually have a placebo, like a sedentary placebo control, or should we actually have an active active placebo control? So should we be comparing rapamycin to exercise, since that's kind of the standard best bet of improving a variety of different markers and indices of health span?
Yes. And that's quite a valid question to ask. I have read, for instance, with regard to metformin, that metformin can mimic the effects of exercise, but if you are already doing exercise, you may be receiving the same effects already as you would from metformin. So it would be very interesting to have an active control group that does regular exercise, and then people on metformin who also do regular exercise, and see if there's any difference. Do you foresee there being any difference with regard to rapamycin between those who exercise and don't take rapamycin versus those who exercise and do take rapamycin? Obviously, the exercise would be at some sort of significant threshold where people are, for instance, endurance athletes, so that it's not just taking a short walk every day.
Sure. Well, I think I can speak on a couple of things. First, I'm going to hit the metformin area first, because we have a study that we were published in Aging Cell, and then Charlotte Peterson's group down at University of Kentucky also published a paper. Ours was aerobic or cardiovascular exercise training, somewhere around 70% of VO2 max, and then the Charlotte Peterson's group was resistance or weight training. And both showed that metformin actually inhibited many of the positive benefits that exercise provided. So it attenuated the improvements in VO2 max. It completely prevented the improvements in insulin sensitivity when metformin was added to exercise. When metformin was added to resistance exercise training, it attenuated the gains in muscle mass and muscle function. So I would say that keep this in mind, that was taking metformin daily. And so I think there's a lot of dosing related questions there. For rapamycin has been used to study how we can inhibit these hypertrophic signaling pathways, and how that it actually has been shown to prevent muscle hypertrophy in response to resistance exercise training. This is when rapamycin is given on the same day of exercise, and this has traditionally been only in young animals. And so I think there's a lot of further research and scientific inquiry in how we can combine these kind of health span promoting pharmaceutical options with exercise. I think there's a way that we can do it, but we so far don't have any great data to suggest that we can.
Yes, this is quite interesting.
I think we should do it. My question, I mean, what is you think should be the ethical stance of, like, can you just be a slob on the couch and take a pill and be good? Or should you do some exercise? You know what I'm saying? I mean, what should we shoot for here? I mean,
Yeah.
Just curious.
Well, I think there's some interesting questions there, right? That, go sort of far beyond the scope of just thinking about aging research. Like, if we're really concerned about climate change, right, then, should we be exercising, right? Like, it costs money to produce the exercise equipment, probably a lot of carbon there, probably a lot of energy to run it if you're running a treadmill.
Go outside.
Doing those things. You're even producing more CO2 while you're running. So if our primary focus is climate change, then we shouldn't be exercising. We should try and have low carbon energy, neutral issues. So I think you view that a lot of different ways.
And of course, from my standpoint, as both a transhumanist and a humanist, my primary focus is on human well-being, human longevity. Now, I think technology can solve problems like climate change, but in order to have technological innovation, we need to have healthy humans who innovate. And so I would be in favor of any habits that maximize human health and improve human health. Now, the reason why exercise is ethically salutary is because it does improve health. Now, if there could be another way to improve health, another way to achieve the same kind of result, then I don't see necessarily why it would be ethically inferior to exercise. But I do think what Adam explained raises some important questions to longevity enthusiasts in terms of what set of approaches are they interested in, and are those approaches actually complementary, or do they inhibit one another in some way? So it seems, in terms of metformin and the benefits for metformin, right now, a lot of the studies, pretty much all of the studies that I'm aware of, have focused on older individuals. So I'm not aware of any studies on younger individuals or younger individuals who exercise. Of course, there's more of a statistical association between being younger and exercising more often for some fairly obvious reasons. So this is an important question for those longevity enthusiasts who are younger and who do perform regular exercise as to what will benefit them, or what might actually offset some of the beneficial effects of what they're doing.
Yeah, I do just to sort of, jump in, right? It's very clear that exercise has all sorts of benefits that go beyond any effects on longevity, right? There's all sorts of positive associations for health and vigor and even, mental performance. So there's tons of reasons to exercise, but 70% of the American population is overweight or obese. So it's very clear they don't have the exercise habit. And I think, no matter how much you try and instill the exercise habit, there's going to be a very large percent of the population who doesn't get it.
Yes. And you could make the argument that could you find a longevity drug that would minimize some of the barriers to exercise for people, right? Do they feel better when they exercise? Does it make exercise a little bit easier for them? The possibilities are kind of endless here. I think this is a really rich area where we can really further investigate what's going on with the combination of rapamycin, metformin, and exercise.
Yes, absolutely. So we have 5 minutes remaining in our virtual enlightenment salon. I wanted to ask Ben if he had a final question for our guests.
I'd like to know why you guys both decided to study aging. What drew you?
Yeah, I guess I can go first. So my background is, so I don't know if you picked this up. My background's in exercise, and exercise physiology, and so I was an athlete, and I wanted to study in grad school. I wanted to study how I can improve people's athletes' functional performance, and I quickly realized that, as it's interesting as that was, I think there's actually a greater impact on society to study how we can improve older adults and how we can improve their function. It's the same goal of improving function. It's just a little bit of a different starting point. So that was really motivating to me. And then I'm sure we probably all have a figure like this in their lives, but my grandfather just always kept telling me, "Don't ever get old, Adam. Don't ever get old." And that was also very motivating to me, and I got to study the science behind it.
I did come at it from the exercise perspective. I came more from the scientific perspective where, when I was starting to go into graduate school, there was a lot of interest around the idea that genes could regulate aging, which once upon a time, was not a commonly held belief. It was commonly held that, genes probably wouldn't regulate aging. And at the time I entered the field, it had just been a few years since genes in C. elegans and yeast had been shown to regulate lifespan. And I thought that was a really interesting field to work in. And as I progressed through my postdoc, I decided that, of all the various fields that I could, continue to work in, this was sort of the one that had held the most interest for me and I could see, doing long term. And I sort of moved into mice and starting to touch on humans now, just because I think, it's so important to be able to bring this out of the laboratory and, move it into something that, can be used at the geriatric bedside, if you will.
Yes, thank you. And it's always interesting to hear the origin stories of researchers in this field because we need more researchers, definitely. In the past, many eminent figures within the field have been stating that funding is the major bottleneck, and funding is still a constraint, as you discussed, but another major constraint that has been mentioned with increasing frequency is the need for additional research talents. So getting more people interested in the field, getting them involved. So in the 2 minutes we have remaining, I would like to offer each of you the opportunity to make some closing statements and provide any other remarks that you think would help round out this session.
Well, I think this is a terrific session, and thanks for inviting me, and thank all the panelists and others for their questions. This was, I think, a really useful discussion as well, and I think there are a lot of great ideas here. I think the key thing is, it's clear that it's becoming a time where mTOR inhibitors and certainly other interventions like metformin are, going to be tested in the clinic. And I think that's really important. There are a lot of other groups also doing trials for various age-related conditions. And so hopefully we'll be able to enroll lots of people at different places and test these different effects of rapamycin, other geroprotectors, and really find something that can extend human health span and hopefully lifespan.
Excellent. Thank you, Dudley. And Adam?
Yeah, I think just kind of echo Dudley's remarks, and I also think that the more we talk about these really promising interventions to extend healthy lifespan, health span, I think we also are starting to understand the more we don't know, and I think it's starting to open up more and more questions that need to be answered. We still do not know the appropriate dosing schemes that we can apply to humans for rapamycin for longevity. We don't know which populations of people may respond the best or favorably to rapamycin. So I think all of these things are just, the more we talk about these and have discussions with groups like you, there's more and more questions that come up that we just don't know. And so I think it's a really exciting time to be in aging research and geroprotective research.
Yes, I completely agree with you, Adam. every time a question is answered, more questions arise. And I think it's quite worthwhile to accelerate the research in this field and hopefully motivate more people to get involved. And I would like to echo the comment made by Jason Geringer in our chat, who writes, I hope you come back and let us know about your progress. We would be happy to have you back for another session, especially once your trial is underway. And in the meantime, thank you so much for joining us today. Thank you to our panel and our very engaged audience as usual. I hope that you found this session to be beneficial in terms of your personal knowledge of the cutting edge of research into aging and longevity and some of the approaches to possibly ameliorate aging. And in the meantime, I hope that all of you live long and prosper.